Hello everybody and welcome to Bite Size Biology, and today is going to be the first of our videos for plants. There's about 5 plant videos going to be made, and we're starting with plant structure. So the main questions that you could be asked in this chapter are to draw or label diagrams of the following: of a root, the stem, monocot and a dicot. You could be asked for the root zones, phloem and xylem, as well as xylem, the vessels and tracheate, which are the subdivisions. In addition to that, you're going to be asked to compare and contrast monocots and dicots. This is a standard enough question, and you could be asked for at least up to 3 different differences, I suppose, between monocots and dicots. You should also be able to name and state the functions of ground, dermal and vascular tissue. State the functions of different parts of the plant. That's fairly standard enough. And there's an experiment involved in this chapter too, and that is to prepare and examine the TS transverse section of a dicot stem, and a dicot being a stem being the choice. That's important, and I have that underlined, and I'll get to that later. Okay, starting off, it came up fairly recently in 2020. Now, the root zones have never really been asked that often, but in 2020 it was asked, and you're asked to answer the following questions about the plant root. Draw a longitudinal section of a typical root, label any 3 zones in the diagram, and in which zone is water absorbed from the soil. So look, I have a diagram there already, and you can see I have 4 different things highlighted. You will need to label 3 zones. I have 4 zones there. We have the bottom, our protection, because the meristem here is where cell division takes place, meristematic zone, and that's very, very delicate. And it does need protection as the roots are growing through the soil in case it bumps against any pebbles or stones or whatever. You then have the zone of elongation, which is over here, and then you have the zone of differentiation, which is over here. So water is absorbed in the zone of differentiation because the root hairs are present, which you can see here, the root hairs. You have a little tongue type thing sticking down here, and that's your xylem and phloem. The xylem is the green part. It's always the part that's on the inside. The phloem is on the outside. So that was the first time in a long time, I think, that this has been asked in a state exam. It's come up a few times in pre-papers, alright. So 2019 also came up, but the experiment came up instead. So what is meant by the term dicotyledous? Well, that just means 2 seed leaves. Don't need to overcomplicate this. Name the plant that you used. Well, there's so many different plants that you could use. Okay, my class used celery, but there are other ones that you could have used. Okay. Why is it important to use a very thin section when examining the tissues of the stem? To allow light to pass through. Now that question can come up for lots of different things. Same for the cell structure experiment. Why must you use a very thin section? To allow light to pass through. Describe 2 steps you use to cut the stem safely. Okay, so you use a scalpel and you cut away from the body. Now there probably are other ones you could say for that as well. Maybe use garden gloves or something along those lines. But they are the 2 obvious ones I put in. How did you transfer the section to the microscope slide? Well, we use the tweezers, but you see some people use paintbrushes with a little bit of water on it. There's probably a few different ones there too, you could say as well. There is actually a few different ones you can say. But tweezers is kind of a standard answer for that. Name the part of the microscope you use to bring the section into sharp focus. So that's the fine focus wheel. Okay, and I'd actually nearly put in the wheel part there actually. So the fine focus wheel. And then draw a label diagram of the section you observed under the microscope. Okay, so this is what you would see. You have your circle, which is indicating it's the stem. It's a dicot, so you have your vascular bundles in a ring shape, which are consisting of xylem and phloem, which I have over here. And then ground tissue is for everything else. So yeah, that was a handy enough one to get full marks in. Oh. Okay, 2014. Now it has actually come up. I know there's a big gap between 2019 and 14. It did come up in other years between then, but they only came up in tiny pieces that I thought wasn't necessary because they're going to only be repeated on this. So next one was draw a transfer section of a young dicotyledonous stem. That's grand, you can see it here. It's the exact same as what you've seen there in the experiment question. Indicating in the diagram the following. So yeah, you had to indicate where they all were, okay? The epidermis is dermal tissue, so I would just refer to that as dermal tissue, really. And I would also refer to the xylem and phloem as vascular tissue, just to be sure. Now, which of the above tissues has a different location in a young root? So you need to know the diagram for a root. Now, the diagram for a root doesn't matter whether it's a monocot or a dicot. Okay, there's just one diagram. Okay, and the answer is xylem. Okay, because the xylem in a root is in the center here. Where previously is the tissue found in the root? Okay, I have it here, xylem in the center, and give one function of ground tissue. Now, you do actually need to know two functions of ground tissue, because this has been asked before. They said ground tissue is responsible for photosynthesis, helps carry out photosynthesis. Name another function of ground tissue. Okay, so you do need to know two. So storage, and make sure you say storage of what? Food. Okay, and photosynthesis. Now, part 3. Draw a label diagram to show the detailed structure of two vascular tissues of plants. Now, if it's asking for two vascular tissues, it wants one to be xylem and one to be phloem. Okay, so I have it here. Now, this phloem here, you can see, was asked in 2017. Okay, as a standalone question. I left it off doing it because we're doing it here. Now, for xylem, I have two versions, which you can see here and here. It doesn't matter which one you pick, but you had to do one of xylem and one of phloem. And that's all you need in it. Like, you don't need to do anything complicated. I know the diagrams in the textbooks and notes and stuff like that could look very difficult to draw, but like really you just need to draw a tube with kind of a snake type figure in the middle of it for the xylem. And the difference between a vessel and tracheid is tracheoids are tapered, so T for tapered, which you can see there, where vessels are not. And phloem, you just have to make sure they have the companion cells in it with nucleus. Okay, I would recommend practicing those. Which of the tissues is composed of living cells? Well, that's the phloem, because companion cells have a nucleus, so it's living. Xylem is considered to be dead. What's the function of the meristematic zone? The meristematic tissue? Well, that's cell division, and specifically it's mitosis. And happens in the root tips and the shoot tips. We'll come across that in another chapter. Okay, the diagram here shows a transverse section of the stem of a monocot. And you can see the difference between a monocot and a dicot. A monocot, the vascular bundles are scattered. In a dicot, the vascular bundles are in a ring shape. What is meant by the term monocot? And you can see there, I made a slight mistake there. I said 2 seed leaves. It should actually be 1 seed leaf. Mono meaning 1. Okay. Give an example of a monocot. Grass. It's the easiest one to go to. Name the structures labelled A. Okay, and that's the vascular bundles. Okay, now it says structures. Okay, so vascular bundles is fine. If you wanted to say xylem and phloem with that, do, but vascular bundles was what they were looking for. And remember, you're going back to 2014, 12, you're going to see the marker schemes aren't going to be as detailed. How do you know from diagram that the section is taken from a stem? Well, there's more than one vascular bundle. So if it's from a root, you're only dealing with one key section there. And how do you know it's a monocot? Well, I said scattered bundles, scattered vascular bundles. How are veins arranged in the leaves of monocots? Well, they're parallel. Think of grass. It's pointy shape. It's very narrow and pointy. Okay. And how does the veins differ in dicots? Well, for dicots, they're going to be netted or branched or something along those lines, which you can see here. So I did say you do need to know the differences. Again, I would know at least 3 differences because they specifically ask about 1 or 2 of them. Okay, we're nearly done. 2010, this kind of comes up in a different chapter, but it's also related to plant structure. So we're actually going to have to see this question again. Just give a roll for each of the following parts of flowers. The sepals, the anther, the stigma. Okay, so you'll see this in plant reproduction. The sepal protects the flower, the anther produces pollen, and the stigma will trap that pollen. Okay, so if you haven't done this sexual reproduction in plants yet, okay, so don't worry about this section here because it'll come up there too. Now, 2008, this hasn't been asked in ages, and you were asked to draw a large table diagram of a transverse section through a young root. Okay, so this is what you have to do here, guys. You drew your circle again, you do your root hairs coming off it, and then in the middle you drew another circle with a star shape. That's how I did it. And the xylem is in the centre, remember I said that earlier, and the phloem is next to the xylem, which is here, 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 and here. Okay, you don't need to be colouring it in. Okay, there's no marks for colouring it in. Okay, there are marks, however, for labelling. Okay, so make sure you have your dermal tissue, your ground tissue, your xylem, your phloem, and root hairs if possible. And finally, 2007, you're given two diagrams here that forms of a vascular plant tissue as seen under the microscope. Name this vascular tissue. Well, there's only two forms of anything that has to be xylem. Now, you can see they look very different to that of your notes, past Markham schemes and whatnot. Okay, this is 2007, however. Identify the two forms of vessels and tracheids. So vessels, you can see tracheids is T for tapered. So if there's a point in it, it's going to be a tracheid. The walls of A and B are reinforced with a hard material. That's lignin. Where precisely this is found in a young stem of a dicotyledonous plant. Okay, so again, I'm not reading the question correctly there. It said stem. Okay, so it doesn't have to be the centre there. That's if it was a root. Okay, it'd be scattered. A ring shape, I should say. So a ring shape. And the reason it's in a ring shape is because if you read it, it says a dicot. Ring shape, okay? And name another vascular tissue, and that's phloem. Okay, so look, that slight mistake there indicates why it's so important to go back and reread the question. Okay, so many marks are lost because students, teachers, whatever, do not read the question properly. So this is what we have here. Now you can see it doesn't come up very often, and when it does, it's actually quite good to answer. I know students find plants boring, but they don't change. The questions are fairly straightforward, okay? And this does link into other chapters, okay? Plant transport specifically, and a tiny bit of plant reproduction. Okay, everybody, look, I hope this helped with your studies, and all the best.